Good day everyone. I am the discussant for this morning. So my topic or my assigned topic is all about a bacteriophage genetics, cell structure and function. But before we go into our discussion, let me tour you first in the world of microorganisms. So, okay. The first topic that I will be conversing is all about bacteriophage genetics. So, we talk about bacteriophage. It is also known informally as a phage. So, it is a virus that infects and replicates within bacteria and archaea. So, the term was derived from bacteria and the Greek phagen meaning to devour. So bacteriophages are composed of proteins that encapsulate a DNA or RNA genome and may have structures that are either simple or elaborate. So their genomes may encode as few as 4 genes, that is MS2, and as many as hundreds of genes. Bacteriophages has its general characteristics. So, the first step toward understanding bacteriophages is to learn something about their classification. However, characterization and classification of the bacteriophages or bacteriophage viruses poses many problems. From a practical point of view, they can be distinguished on the basis of their natural host range, and other similar characteristics. In addition, they can be characterized on the basis of their RNA or DNA contents. So each phage generally contains only one kind of nucleic acid, that is the filamentous viruses have been shown to be an exception on this general rule. So the structural symmetry and susceptibility or resistance to ether and other solvents and to other chemical agents also provide additional criteria for the classification. So as you can see in the table 6.1, that is the characteristics of some phages of E. coli. So we have phages, the virion morphology, nucleic acid type and amount, latent period, the average yield per cell, and the growth cycle at 37 degrees Celsius. So let's have, for example, for T1. So the virion morphology of it is what we call icosahedral, having the measurement of 50 nanometer of its head, and the tail is 10 times 150 nanometer. So the nucleic acid type of amount is the DNA having 2.5 times 10 raised to positive 7. The latent period is 13, and the average yield per cell is 150. So, the icosahedral 50 can resist to drying. Bacteriophages range in size of 20 to 300 nanometer. So large phages are barely within the realm of resolution of the light microscope. For visualization of any details of viral structure, the electron microscope is essential. So the nucleic acid may be either double-stranded, single-stranded, circular, or linear. So morphologically, phages generally display a considerable degree of geometric symmetry. So the general have a head or capsid composed of a identical protein subunits called capsomeres. The nucleic acid core is housed within the capsid. So some bacteriophages have a prominent tail structures. So the capsid structure may assume in icosahedral form of 20 sites within 12 vertices. So the entire infectious unit is generally referred to as virion or virion. So the smallest phages contains nucleic acid, either DNA or RNA, with a molecular weight in the order of 1 × 10 raised to positive 6 Daltons. So this relatively short chain nucleic acid can obviously only code for a small number of genes. So these genes of necessity must encode information that governs the formation of the basic viral subunits. So the presence of such a small number of genes reflects the degree to which viruses are dependent on the host cell. Each species of microorganism is susceptible to a limited subset of viruses. So put another way, each phage has a specific and limited host range of susceptible microorganism that it can infect. For example, a hypothetical phage A might only infect one strain of E. coli, while phage B could infect many different strains of E. coli. So phage A is viewed as having a narrow host range relative to that of phage B. Host range depends on the presence of a specific viral receptors on the host cell surface. So these receptors are usually composed of specific carbohydrate groups on lipopolysaccharides, or what we call LPS, of cell surface structures. So some viruses such as M13 attach to 6 pili and are referred to as male-specific phages. So the salient point here is that receptors are actually normal cellular proteins with specific functions that are co-opted by the phage. Bacteriophages or bacteriophage infections sa bacteria follow a characteristic pattern. So the growth curve of bacteriophages, sometimes referred to as the one-step growth curve, is depicted in figure 6.1. So once a population of bacterial cells has been inoculated with a given number of bacteriophages, the number of detectable infectious particles rapidly decreases. So this stage is termed the eclipse phase and represents that portion of the time following infection during which bacteriophages cannot be detected either in the culture medium or within the cell. So the overall period encompassing absorption and eclipse is referred to as latent period. So after the latent period has been completed, approximately 20 minutes for E. coli, cells infected with coliphage, infectious viruses began to be released from the cell. So the average number of infectious bacteriophage particles released per cell is referred to as the burst size. So the replication cycle may be depicted as follows. The first is the bacteriophage attachment to cells and pili, as you can see in figure 6.2. So the subscript letter A and B, that is the attachment of T-even virions to points of adhesion between cytoplasmic membrane and outer membranes cell wall of E. coli. E. coli bars equals to 100 nanometer. Then the E. coli cells with F. pili showing the attached particles of M2, that is the icosahedral, and M13, the filamentous phages. So the second replication cycle is what we call the injection of nucleic acid or penetration of the entire virus in the cell. So then, uncoating, that is the removal of the capsid surrounding the nucleic acid or the core. Then the intracellular synthesis of viral subunits or the capsid and nucleic acid. Then assembly of the subunits into complete virus particles or naturation to complete the bacteriophages within an E. coli cell are as seen in figure 6.3, a section of a cell of E. coli strain B from a culture infected with bacteriophage T2. So clearly visible are the cell envelopes, the walls and cytoplasmic membrane, the clear area of the phage of DNA pool containing many condensed phage DNA cores, and attached to the cell surface, 3 phages, 1 empty and 2 still partially filled. The phage at the top shows the long tail fibers and the spikes of the tail plate in contact with the cell wall. So the tail's sheath is constructed or contracted, and the tail core has apparently reached the cell surface but has not penetrated it. So we come now to the T4 phage. So the T4 phage is one member of the Myoviridae family of T-even, the T2, T4, and T6, like tick phages of E. coli. So as you can see in figure 6.4, so that is the schematic representation of phage TB4 virion. It is its component part and penetration of the phage core through the bacterial envelope. So the subscript A, virion, is the representation of virion with extended tail fibers, while the subscript letter B is that the virion with the tail sheath contracted and the spikes of the tail plate pointing against the bacterial cell wall. So the TB4 or T4 possesses an oblong head having the measurement of 80 times 120 nanometer and a contractile tail having the measurement of 95 times 10 nanometer. So within the capsid is linear, double-stranded DNA molecule. So having the measurement of 1.3 times 10 raised to positive 8 Daltons or 1.69 times 10 raised to positive 5 base pairs. So associated with DNA are the numbers of polyamines or polyamines, putrescine, spermidine, and cadaverine. So the base composition of T4 DNA is unusual in that all of the cytosine residues have been replaced by a modified base. So hydroxymethylcytosine or the HMC, the reason for this replacement will become apparent from subsequent discussions. So while virions in general are thought not to possess enzymes, a number of enzymic activities that have been found to be associated within the TV4 or T4 virion. Several of these are listed in table 6.3 along with their possible rules in T4 infections. So, we come now to the general pattern of T4 gene expression. So, in early gene expression, it is general strategy of viruses to differ some gene expression to later stages of infection and progeny virus maturation. So, the T4 phage is no exception. So, transcription of certain T4 genes begins almost immediately. So, the immediate early genes are transcribed at 30 seconds post infection, and the delayed early or middle genes are transcribed at 2 minutes post-infection. So as you can see in figure 6.5, that is the life cycle of T4 phage. So number 1, that is the attachment and injection of DNA. Then the number 2, transcription of early genes. And number 3, that is the replication and concatemer formation. And number 4 is the transcription of late genes. Number 5, that is the head assembly. And 6, tail assembly. And number 7, that is the attachment of head to tail. Then number 8, attachment of tail fibers. And number 9, cell lysis and release of mature phage. Okay, so the products of these genes establish the infection, since transcription of the immediate early genes is described by a lambda 70 promoters. A normal host RNA polymerase is used. So among the early gene products are nucleases and other T4 proteins that stop host transcription, unfold the host chromosomes, and degrade host of DNA to nucleotides. So several enzymes are involved with the synthesis of T4 DNA, including the T4 DNA polymerase. Okay, so we come now to the middle gene expression. So the transcriptional switch from immediate early to middle genes is associated with series of T4 directed modification as shown in table 6.4. So the ALT and MUD or MUD proteins catalyze the ADP ribosylation of ARG265 and RNA polymerase of alpha subunits. So this modification lowers the affinity of core polymerase for the sigma factor with a molecular weight of 70 protein, and is part of a strategy to reduce host RNA synthesis. So the ALT protein is actually packaged in the T4 capsid and injected with a DNA during infection. So the MUD, on the other hand, is synthesized after infection. So the transcription of middle genes requires the motility protein A, a positive regulator that binds to a TAT or the EGCTT. So EGCTT, DNA sequence of 13 BP upstream of a negative 10 box. So converting the region into an active promoter even though the region lacks a negative 35 box. So T4 also produces a 10 KDA or anti-alpha 70 protein that prevents transcriptions of the T4 sigma factor of 70 early genes promoters. So the ACA has been shown to bind region of 4.2 of sigma 70. So blocking interaction between the sigma 70 and the negative 35 upstream promoter element of early T4 genes and most E. coli genes. So let's come now to the late gene expression. So expressions of the early and middle genes ceases about 12 minutes post-infections. So however, at 5 minutes post-infection, the DNA replication has begun and transcriptions of the late genes begins. So further modification of RNA polymerase accompany this transition. For example, the JP55 is a viral encoded sigma factor that is required for the late gene transcription. So it is essentially hijacks that the host RNA polymerase by altering its promoter sequence recognition properties. So this works because the molecular structures of T4 late promoters are quite different from other non-prokaryotic promoters. So it uses the consensus sequence of TAT, AAA, TAC, and TATT. So at the 10 negative region but exhibits no consensus sequence at the negative 35 regions. So the alternate sigma factor of GP55 and the unusual structures of late gene promoters directs T4 transcription toward the late genes. So another curiosity of T4 transcription is that expression of late genes is closely tied to DNA replication. So one of the proteins involved with late genes transcription of GP33 serves as a link between the RNA polymerase of negative sigma GP55 and components of the T4 replication machinery. So apparently, the T4 replication complex serves as the mobile enhancer, although how this works is not clear. So we come now to the T4 genome. So terminally redundant and circularly permuted. So it should be noted at this point that while the DNA of T4 phages is linear both in the virus and within the host cells, the genetic map is circular. So that is situated at opposite ends of the linear structure, which should not map close to each other. So nevertheless, upon genetic mapping are found to be linked. So the resolution of this paradox can be found in the realization that the ends of each T4 DNA molecule are terminally redundant. That is, a duplication of genetic information occurs at both ends of the single molecules. So this phenomenon is the result of the heedful mechanisms utilized for the packaging of T4 DNA into the T4 capsid. So after DNA replication, replica molecules can recombine with each other at their terminal redundancies, creating very long molecules, many genomes in length. So each mature virion is then made by packaging a headful of DNA cut from such a T4 concatemer. So one headful, however, is 105% of a single genome. Therefore, the packaging process will generate a population of phages carrying all possible circular permutations of the T4 genomes, as you can see in figure 7. So genetic crosses between members of this population will result in a circular map. The T4 DNA replication, so there are approximately 30 T4 genes known to be involved with some aspect of T4 DNA synthesis. So many of these are not directly involved with the replication fork mechanism but encode enzymes required for nucleotide precursor synthesis. So in this section, we deal only with those genes directly involved with the replication process as well as the analyze the replication machinery itself. So because of its rapid infection cycle, T4 uses multiple replication origins and initiation mechanisms. So there is an origin-dependent early mode of initiation involving multiple origin space around the T4 genomes. So transcription in these areas is essential to initiating replication. So as you can see in illustrated figure 6.8, so transcription, it at an origin creates a substrate for RNase SEH following cleavage of the RNA and the RNA with the ratio of DNA hybrid. So the remaining RNA serves as a replication primer. So one major initiation site occurs between genes R2 and 42. And there are up to 4 additional sites at other locations. So, as with host DNA replication, there is a leading or continuously synthesized strand and a lagging strand. So, Okazaki fragments in T4 are about 2,000 nucleotides long. So, the major protein responsible for nucleotide synthesis is GP43 and the T4 DNA polymerase. So, having the MWT 110,000. So, accessory proteins include G44 to G45 and G62, which collectively form a sliding clamp on the DNA much as the DNA AB protein does on chromosomal DNA. So, regulation of the T4 gene expression. So, regulation of T4 gene expression has been shown to occur both the transcriptional and translational levels. So, at the translational level, two systems are recognized. So, autogenous translational reparation by the gene 32 product, helix destabilizing protein, and the translational reparation of many T4 induced early mRNAs by the phage Reg-A product, and the system of translational reparation already discussed. So each protein repressor was specific to only one species of mRNA. So this mechanism appears the same for the T4 gene 32 product. However, translational regulation by Reg-A is unique in that it appears to repress the translation of different mRNA species. So the mechanism appears to be the simple recognition of unbinding to consensus mRNA sequence of AUG, UACAAU, -3. So thus blocking ribosome access. Secondary mRNA structures appears to be unnecessary. So although the regulatory significance of Reg-A remains a mystery, so it would seem to play a role in coordinating the early events in T4 development. So we come now to the LAMP phage. So as you can see in figure 611 of E. coli, is a Cyphoviridae and a classic example of temperate phage. So that is, it undergo either a lytic or a lysogenic cycle upon infecting a sensitive E. coli. So this property manifests itself in the form of a cloudy plaque when a lambda is grown on the lawn of E. coli. So the cloudy center is due to growing bacterial cells that have been lysogenized by the phage DNA and to the fact that these lysogens are resistant to superinfection. So mutations in the certain lambda genes will prevent lysogeny so that the mutant phage will be produce clear plaques in which all the infected cells are lysed. So the lambda genome having 42.5 kDa expresses approximately 50 proteins. So the genes of this phage can be grouped into 4 categories based on the rules in the either lysis or lysogeny. So first, genes involved in the lytic development, and the second is the genes involved with the development of lysogeny. So the third, genes participating in both, and the fourth one, genes with the unknown functions. So the genes in the second and fourth groups are non-essential for the phage growth, as you can see in figure 612a, present a general map of the lambda genome. So the DNA within the lambda capsid is linear 48,502 BP but contains cohesive ends designated to cosine sites. So upon phage attachment and injection, the cosine ends of lambda DNA anneal and are ligated to form a circular molecule. So there are two phases of lambda replication as the phage undergoes lactic development. So that is an early phase in which the circular molecules replicate bidirectionally, and late phase. So approximately 10 minutes post-infection, consisting of a rolling circle mechanisms that generates long concatenameric molecules containing multiple copies of the lambda genome. So this discussed in details below. So the concatenamers are then acted upon by the lambda packaging system that are used to fill preformed capsids with lambda DNA. So a precise amount of lambda DNA is packaged corresponding to one complete genome following cleavage by the lambda terminase at the unique cosine sites mentioned above in figure 6.13. As you can see in figure 6.13, that is the infective pathway of bacteriophage lambda. So the phage DNA forms as covalently closed circle through pairing of the cohesive ends, then followed by the ligation. Early DNA replication by the mood generates circular monomeric molecules. So the switch to the rolling circle mode of the replication is dependent on the product of the GAM genes, which inhibits the host RIC-BC nucleus, and the multimeric DNA is the substrate for encapsidation. So we come now to the lysis lysogenidization. So upon infection of lambda phage must make a molecular decision to multiply in a lytic manner or hibernate. As an integrated prophage in the lysogenic state, as will become evident, it is important for the lambda to go down one path, that is toward lysis, without partially going down the other, that is toward lysogeny. So the multiplicity of infection greatly influences the final decisions. So in general, a high multiplicity of infection leads to lysogeny, whereas a low ratio of phage to cells results in lytic multiplication. To understand better about the transcription of how lambda initiates the leftward promoter and the rightward promoter, here is the video.
So this is the bacteria right here, and this is the chromosome of the bacteria, and this would be a bacteriophage. This is the virus that attacks the bacteria. So if we kind of see what's going to go on here, this particular virus is going to, as you can see, inject its DNA into the actual bacteria. And so now this bacteria has its own DNA, and it has the DNA from the actual bacteria. And so if we see what happens from this point on, we'll now see that the host DNA has been destroyed. The base of the phage DNA, the genes in the phage DNA directed the host DNA to fall apart. And so now the only DNA that's left in the actual bacteria is the DNA from the virus. And I'm sure you can figure out what's going to happen from this point on. Now, of course, the ribosomes of the bacteria are going to be producing proteins. These proteins are viral proteins. And as you can tell here, the phage DNA is going to now code for the production of more viruses, and of course now these viruses are going to at some point explode from the cell, and that is basically the lytic cycle. And of course lytic means to lyse, or it means to cut or break open. So this results in the destruction of the bacteria, and that is basically the lytic cycle. So now we're going to go through the lysogenic cycle. So now this is the lysogenic cycle, and this differs in a couple ways, and I'll talk about them after. So the first part of this is the same, that basically a virus is going to attack the bacteria. And so what they didn't show here is that the virus would have landed here, it would have incorporated its DNA exactly the same as it did in the lytic cycle, but here's now the difference. In the lysogenic cycle, we see that instead of the DNA from the virus destroying the host DNA, it actually integrates itself into the chromosome of the host DNA. So now the difference is that, there is no destruction yet. So the beautiful thing for the virus is that, well, basically, it's not going to be making a lot of its babies, for lack of a better word, or offspring immediately, but this cell is going to start to divide, and it's going to divide zillions of times. And every time it divides, you can see that little teeny piece of DNA from the phage is going to, it's basically referred to as prophage DNA, is going to be carried from generation to generation. So now you have potentially millions, if not hundreds of millions of bacteria that all have this little piece of viral DNA. So at some point, when this host cell becomes either stressed or exposed to ultraviolet light or some kind of chemicals, a system called phage induction happens. And basically what happens, you'll see here that that little piece of viral DNA pops out, and now basically the cell goes through the lytic cycle. So the second half of the lysogenic cycle is basically the lytic cycle. So what's going to happen? It's going to destroy the host DNA, it's going to start making the proteins, and there we go. So the difference between these systems is that in the lysogenic cycle, the chromosome or the nuclear DNA material from the virus incorporates into the actual phage DNA and is carried generation to generation. So the bacteria doesn't make it, or the virus does not make as many offspring as quickly, but it is incorporated its DNA into many, many more cells, and thus giving the ability to potentially make much more offspring over a longer period of time. And so that is basically the difference between the lytic and lysogenic.